Okay, everybody. So welcome to this very first of our virtual series for Meeting a Scientist. We're very, very fortunate to have Dr. David Jackson here from Cold Spring Harbor Laboratory, who we're just going to introduce in a moment. Before we do, let me just share some things with you. Number one, this is designed to be an interactive session. So at the bottom of your screen, you're going to see that there's a place called Q&A. And we invite you through the presentation to ask any and as many questions as you can. I think that that's what our speakers like and enjoy, interacting with students. So that's something there. One other thing, I'm gonna actually share my screen for a moment. So I'm gonna stop the slide up there, Dave, for just 1 second. And I'm just gonna share the DNA Learning Center page. Wait, let's see, am I sharing everything? Hold on 1 second. There we go. I wanted to share just this one page. Hopefully you've visited the DNA Learning Center website. Maybe that's where you've gone to register for this event. You got an email. But I wanted to remind everybody that if you come up to our Fall 2020, or if you look at our Fall 2020 programs, there are a whole bunch of virtual programs that are going on. Many of those, some of them are in school or some of them are at home. Many of them involve a kit that you can do stuff at home. So if you are not able to go into school at the moment, you can actually still participate. So take a look there, or if you're a teacher, take a look there, or let us know if you have any questions. Regarding this series, in addition to David Jackson, we're going to have on December 10th, Dr. Camilo dos Santos, and that'll finish up us for right into 2021. Lloyd Trotman will also be one of our speakers, and we actually have just about the whole school year planned out. So please pay attention to what's coming up and share this and circulate it. We'd love to see everybody attend these and find something interesting. So with that said, I'll remind you again, you can go ahead and put your questions into the Q&A. But I'm about to turn over to Dr. David Jackson, who's a professor here in the plant sciences group at Cold Spring Harbor Laboratory, and he has wonderful stories to share. So we're going to give him his attention and allow him to go ahead and start. Thanks very much for joining us, Dave.
Okay, thanks very much, Jason. Can you see my slides okay?
Yep, everything is
okay,
there.
Great.
Turn my video off and sit back and watch.
Yeah, and thanks everyone for joining. I'm really happy to have the chance to be able to tell you about a little bit about my story, how I became a scientist, and also a little bit about our recent work. And it's really a story about how what we call basic research, fundamental research, research that you might not think would have a serious application, really could be used to tackle real world problems, such as improving agriculture. So I'll get to that in the second part of the talk. But before that, Jason and the others at the Learning Center asked me to cover 3 things during my talk today. One is how I got to be a scientist. And second is to talk about things I had to overcome to get where I am as a scientist. And then, as I said, I'll finish up by telling you about some of our recent research. So, the story about how someone becomes a scientist, I think, is probably different for everybody. It's a very personal thing. Myself, I think I was probably born a scientist in that I was always really fascinated with the world around me and trying to figure out how things work. I think one of my earliest memories was getting one of my brother's toys, which was a teddy bear that had a music box inside. And I just had to know how it worked. And so one day when no one else was around, I got a pair of scissors and I did surgery on the teddy bear and cut out the music box, opened it up, and inside was 1 million pieces. It was an old mechanical music box. So I took those out and ended up with just a pile of tiny pieces on the floor that I had absolutely no way I could ever figure out how to put it back together again. And so, I basically, no one had seen me, so I hid it under the bed. My brother was very sad for a long time. And I think I probably got into trouble when my mom found out what I'd done. But, that's sort of my childhood. I spent it really figuring out, taking things to pieces, breaking them, and trying to figure out how things work. I grew up in the north of England. My accent has been lost a little bit, but you might spot it. And it's an area of great beauty. And I think that's probably what inspired me to love biology. But also it's an area where the Industrial Revolution started. And actually my father worked in one of these old factories as an engineer and had a small workshop at home. And so again, during my childhood, we spent a lot of time making and breaking things. And that probably also inspired me to love science, which is basically figuring out how the world works. But when I was your age, in high school, what I thought I would end up doing in my life is actually a little bit different from what I'm actually doing now. At that time, I was really excited about this new area called biotechnology. So in the early 80s, people had recently figured out how to clone DNA. And there were some very famous biotech companies like Genentech in California started to be formed. And my dream at that time was to go and work for one of those companies. And so I applied and went to my undergraduate college, the University of Leeds, to take a degree that this university was one of the few offering that at the time, an undergraduate in biotechnology. And that's what I thought I was going to do, work in a company. But when I, in the final year of my undergraduate project, I did a research project in the lab of this professor, Professor Diana Bowles. And it's then that I really realized that I love just basic research, just being in a lab, being able to plan your own experiments, and really no one's telling you what to do. It's curiosity driven, and you just want to find out things about how things work. And so at that time, I sort of had a bit of a moment of indecision. Part of me still wanted to go and work for a company, but also I was very intrigued by this idea of academic research. And so my mentor, Diana Bowles, suggested that I should just work in a lab for a year after graduating. And, I wasn't so sure, but then she said, well, I have a collaborator in Paris. Maybe you can go and work in that lab. And at that time, and still do, I love traveling. And so the chance of going to live in Paris for a year was amazing, I thought. And I almost forgot about science and just went there and had a great time. And during working in the lab there, this is the famous Thinker by Rodin, a statue in Paris. And I spent a lot of time thinking, and I really decided that lab work was what I loved and research was what I loved. So that's what I wanted to spend my life doing. So to proceed in academic science, usually people get a PhD. And so that was my next step. And I went back to the UK and studied for my PhD in a research institute in Norwich in England. And I had two research mentors, Keith Roberts, who's actually a good friend of the lab of Cold Spring Harbor. He's written books with Jim Watson, and Kathy Martin. And during that time, my thesis research was something quite abstract and esoteric, you might say, in that we were trying to figure out why different snapdragon varieties had different colors, right? So you've probably seen these flowers in the forest. There's a lot of variation in colors. And it's really, actually, it's a model system for understanding genetics. Actually, Gregor Mendel also worked with this plant. So I spent my time searching for genes that control these differences. And we found some of them. And it was really fun. But I kind of felt like I wanted to do something a little bit more relevant to agriculture and a bit more applied. And so after a PhD, again, to carry on the academic path, somebody would normally do what's called a postdoc, which is another training period. And I had met a professor from UC Berkeley in California, Sarah Hake, at a conference, and really was inspired by her research and also her style of talking about science and mentorship. She was just so excited about her science, and really that came out when you talked to her. So I was really excited about that. And I went and worked in her lab for 5 years, where I started to work on corn, which is obviously an important global crop, and really trying to understand how the plant grows and develops using genetics. And when you're a postdoc, people say it's the best time as a career scientist, because you've done your PhD, you've learned a lot of techniques and approaches, and you're just free to do research. And it really was an amazing time. I had all the resources I need, nothing else to worry about, just go to the lab and do cool experiments. So it was really great. But that kind of period doesn't last forever. And after the postdoc is when a scientist would in academia would then get their own independent position. And so usually during that time, you collect a research ideas and materials. In my case, because I'm a geneticist, I was collecting mutants, which I'll show you in a minute, that enabled me to set up my own independent research lab, basically to break out on my own and have my own research group, which I have now. And so, yeah, so after my postdoc, I was actually very fortunate through connections to hear about an open position at Cold Spring Harbor Lab, which obviously is where I am now. So I moved there a little over 20 years ago, and it's been really, it is an incredible institute, and it's been really an amazing place to spend a good part of my career. So one of the amazing, great things about science is that you get to work with people from all over the world. So this is pictures of my research group at different times. This one at the bottom is quite old. Again, this is about 6 years old, but you know, there are people from my group who now have their own research groups across the US, in Europe, in Asia. And yeah, so I have a network of friends and colleagues around the world who I visit quite frequently. And that's really, really great. And it's, you know, when you become a professor, or what we might call it a PI, principal investigator, you often lose the bench work. I mean, I don't really work at the bench anymore, but I still have these great connections. And also I get a great deal of pleasure from knowing that the people in my lab are successful and they go on to launch their own careers, which is great. Okay, so that's kind of how I got to where I am. So some things about, you know, what I had to overcome. So what are the hard parts about being a scientist? I mean, there are a lot of them. I think I just point out a few here. So one is that, you know, in the beginning, I had no idea what a career in science would be like, you know, and that's because I didn't know any scientists. And I think it's maybe true for many of you, you know, science is not a very common career. And yeah, it's just not the kind of person you might run into every day. So yeah, so I really was uncertain what it would be like. But, you know, in retrospect, I can tell you it is an amazing career, and it's been really, really wonderful. Another big issue, or, you know, hardship, I would say, not really hardship, but difficult thing, is that when you are training as a scientist, you learn how to do science, you learn how to do experiments. But then when you make the transition to be a professor, then you have to, you know, kind of get away from the science a little bit and start managing a group of people, which, you know, scientists are not really trained in management, so it can cause some issues. I've been fortunate to have really great people in my group and haven't had many issues, but it's something that takes a lot of time, and aligning expectations with people you're working with is something that does need a lot of work. And then the last thing, as a scientist, you have to be self-confident and a little bit tough-skinned, because you deal with a lot of rejection. So we, you know, the currency of being a scientist is that we publish papers, and we get funding by applying for research grants. And without either of those, you can't really be successful. And, you know, both of those areas are extremely competitive. So it's very common that you may spend a long time on a grant proposal and submit it, and some of the granting agencies only fund maybe 15% of the applications they get. So it's very common to get a rejection, and you have to, you know, be positive and, you know, try to take criticism positively and move on. Similar, yeah. But you know, despite those difficult things about being in science, I would say follow your dreams. If you love science, it's an amazing career, and I couldn't really imagine doing anything else. Okay, so now I'll get on to the easier part of the talk, which is to talk about science, what I love. So something I'm excited to be working on and its impact. Okay, so my lab focuses on plant genetics, and we mostly work with corn, which is a very important plant. It's actually the #1 crop globally in terms of total production. So the work is quite relevant, but that's not really the reason I work on corn. The reason is that it's a really wonderful model system for doing genetics. And I'll just show you, you know, people have worked on corn for actually over 100 years. And I'll just point out one example, which is the work of Barbara McClintock, who was at Cold Spring Harbor for most of her career. You probably know about her work. She discovered these things in corn called transposable elements. She actually called them something differently, controlling elements, which actually is a profound difference, because it means she really understood what these DNA elements do. And her work was actually not recognized for a long time, because originally people thought it was something specific to corn, sort of an oddity of corn. But now we know that, for example, our own genomes are made up of a very large proportion of these regulatory transposable elements. And so it was deeply profound work. And because of that, she was awarded the Nobel Prize. So she's a great inspiration to my work. Excuse me. So in my group, what we focus on is using genetics to try to understand how plants grow. So you probably know that there's a very fundamental difference between plants and animals, in that plants produce organs throughout their life cycle. So leaves, flowers, seeds, they're all made continually throughout the life cycle. Whereas in humans, for example, our bodies are formed, and at birth, we really just grow. We don't really make new structures. So if you cut off your hand, then it's gone forever. But you can cut off a leaf or a flower, and they will grow back. So plants have this amazing property of regeneration and self-renewal. And the way that plants do that is that they have populations of stem cells, very much like the pluripotent stem cells that you've probably heard about in humans. So where are the stem cells? Well, if you take a corn seedling and just dissect away all the leaves, you get to a structure that we call the meristem. It's a collection of stem cells that is critical for plant growth. Without this, the plant cannot survive. And this small population, this is just about 1/10 of a millimeter in diameter, but this small group of cells is responsible for maintaining itself as stem cells. And when these cells divide, they make small primordia that will make leaves, and also the other primordia that will make branches. So really, this structure is critical for controlling how the plant will look, what we call the plant architecture, how it will be. Now, one of the reasons we're interested in the meristem is that we think it may have been something that was selected for when crops were domesticated. So you probably know that plants like corn, wheat, tomatoes, a lot of the things we eat did not exist in their present form thousands of years ago. Like 10,000 years ago, we only had wild plants that were very low yielding. So in the case of corn, the ancestor, which still grows in Mexico, is a plant called teosinte. And teosinte ears make only about 10 seeds, whereas modern corn makes many hundreds of seeds. And basically, ancient farmers took teosinte and they selected for higher yielding varieties, which is how we come to have crops today like corn. And one of the things I think they selected for, although not knowingly, is a bigger meristem. So the meristem here is colored in green, and the bigger meristem can make more of these structures, which are the flowers that will make the seeds. Okay. So how did this happen? Well, one way, and actually, not only is there a big difference between corn and its ancestor teosinte, but even in modern corn, there's a huge amount of diversity. So this is just a collection of different ears of different corn varieties. And you can see there's a huge diversity in pigmentation, but also in the size of the ears, in the length of the ears, and the width and the shape. And so what we want to understand is, what are the genes that underlie some of this diversity? And in my group, we studied this by studying mutants. So I think you're probably all aware, a mutant is a plant where a gene is broken, where the gene has been disrupted, so it's no longer functional. And so I'm just going to tell you about one group of mutants we call them fasciated ear or FAIR mutants. And in these mutants, as you can see, the meristem, which is normally a very small organized structure, is massively enlarged. This is almost like a version of cancer in the plant where the stem cells are massively over proliferating, and you get a meristem that's really disorganized and, you know, tens or hundreds of times bigger than it should be normally. And so one thing we can do, and sorry, I forgot to point this out, these images, in case you don't know, these are scanning electron micrographs of the ears at a very early stage. But later, when the plants grow up, the ears of these mutants, as you can imagine, look very disorganized. And one of the things we can do with these mutants is we can find the gene that underlies the mutation, the gene that's been broken to make this mutation. And in this case, this gene encoded a protein, which is a kind of protein that we call a receptor, that sits in the cell membrane and receives signals from other cells that tell the stem cells how to divide. And actually, there are a lot of mutants like this in corn. This is one of the reasons we worked on them, because we had a lot of material to start off with. And we've cloned a lot of these genes and had very interesting stories to tell about them over the years. And if we put all this work together, and what I'm doing now is putting together work from my group, but also work from many groups around the world, including groups in the USA, in Germany, in Japan, China, and many places. We can come up with a model of how the meristem works. So this is, again, the meristem. And what we've figured out is that there are two groups of cells in the meristem. In blue here are what we call the true stem cells, the pluripotent cells. And below them in purple is a group of cells that we figured out only by studying the genetics, is that there's a second group of cells that we call the organizing center. This group of cells sends a signal to the stem cells to instruct them to be stem cells. And then the stem cells send a signal back to the organizing center, a negative signal. And this negative feedback loop is what keeps the meristem in this nice, controlled, regular pattern. Okay, so that's two important groups of cells. And I won't go into all the details, but we know now a lot of the genes that control these different functions. So some of them are signaling proteins, as I mentioned, like these receptors. And some of them are transcription factors, which are obviously important proteins that control how other genes are turned on and off. Okay, and I'll just show this briefly. This is a view of the same molecular pathway, but now showing the molecules. So the receptors, this is a cell, the receptors are in the plasma membrane. They're signaling to a gene in the nucleus, this gene WUSCHL, and WUSCHL is feeding back to activate expression of the receptors. So a lot of these details are very, very clear now and have been worked out over the past 10 years or so. Okay, so what I want to talk about is just one experiment where we've thought about if we can control the size of the meristem to control how many seeds is made by the corn plant. Now, this is now, again, a microscope image of a developing ear. So in the middle here is the meristem. And going off in the background, these rows of bumps that you see, these are rows of actually secondary meristems that will make seeds eventually. And so what we have wondered over the past few years is that if we could make this meristem just a little bit bigger, then maybe there would be more space around it to make more of these rows and more rows of seeds, right? So that's pretty simple. And so, but this meristem is hidden away deep in the plant. It's tiny, it's very hard to access. So how do you make it bigger? Well, we know we can make it bigger using genetics, right? We have mutants, these Phayer mutants that make it much bigger, like this, for example. So we can make the meristem with just a simple mutation. We can make the meristem much bigger, but these mutants, as I showed earlier, are very disorganized, and their yield, the amount of seed they produce, is actually very low. And so what we would like to do is take one of these genes, but not knock it out completely, but just reduce its activity a little bit so that we could go from a situation where we had a small meristem to have a bit bigger meristem, okay, and more space to initiate these seed rows, so more seeds, and ideally higher yield. So we call this, in biology is about balance. We call this the Goldilocks spot, where the meristem is not too cold, not too hot, but it's just right. So we've optimized the size of the meristem to increase the yield of seeds. Okay, so how do we do that? How do we reduce the expression of a gene rather than knocking it out completely? And so I'm sure you've all heard of CRISPR, very famous recent discovery in biology that actually led to the Nobel Prize in Chemistry this year for two amazing female scientists, Jennifer Doudna and Emmanuelle Charpentier. And what they discovered is that bacteria have a system called the CRISPR system, which is able to defend, it's part of an immune system that defends bacteria against invading viruses by cutting the DNA of the virus. And the way that it does that is that there's a protein called Cas9, which is a nuclease. It's an enzyme that cuts DNA. And then there's a second component called a guide RNA that guides the Cas9 enzyme to the position in the genome by matching, base pair matching, that guides it to the position of the genome where you want to make a cut in the DNA. Now, the other thing I need to show you as introduction is just to remind you of the structure of a gene. So genes, as you know, genes are transcribed to make RNA, and then RNA makes the protein. So that part of the gene that makes the RNA and then the protein is what we call the coding region of the gene. But there's another very important part of the gene, which is the promoter. And this is the part of the gene that controls how much of the protein is made and where it's made. So this contains sort of the information about where and when the gene is expressed. So if we want to use CRISPR to modify a gene, one of the very common approaches is you just use it to make a mutation in the coding part of the gene, but that is quite likely to knock out the gene completely and give a complete loss of the function. And so what we've done in this experiment is to design a way to use CRISPR to target the promoter. So we control how the gene is expressed, not whether it's on or off, but by basically, we actually call this fine tuning the gene. And so we've done this in maize, in corn, maize is the same thing, where we use Cas9 and guide RNAs, actually many guide RNAs that target the Cas9 enzyme to the promoter, the upstream regulatory region of this gene that normally when you mutate this gene completely, because of very strong and very low yielding phenotype, but we're targeting the promoter. And sometimes these Cas9 molecules with the guide RNA will cut at the same time, and a piece of DNA between two guide RNAs will be deleted. So we call these promoter deletion alleles. And as you can see, this is on the right here, we're measuring expression of the gene. And in many cases, when we delete part of the promoter, as you would expect, the expression is reduced. So what do the plants look like? Well, they look exactly as we had really hoped they would, in that they are not as strong as the complete loss of function, so they make ears that are as long as the normal corn, but they're wider, they have more rows of seeds. Okay, so suggesting that these promoter deletion mutations might actually increase the yield of the corn plant. And in fact, we've measured that, and indeed we find that with these alleles, we can increase yield. In this experiments we've done up to now, we've had yield increases up to 26%, which is quite amazing, I think. Now, I will point out that in our lab, we don't work with the kind of varieties that farmers grow. We work with sort of lab strains, lab rats, if you will. So it's not clear yet. Well, it wasn't clear up to this point if this would be relevant to agriculture. But I wanted to just show you one result, and this is my last slide, from a former a researcher in my lab who, Son Vi, who went back to his home country, Vietnam, and is now working there as a researcher, as a professor, and also as a corn breeder. And he took some of these weak allele varieties that we'd made and crossed them into elite hybrid varieties that are grown by farmers in Vietnam, and actually showed that in that situation also, he can increase yield in some environments by up to 24%. So this is really, I think, an amazing result that could have a big impact on global agriculture. Okay, and so with that, I'm done. Oh, sorry, last slide. I want to remind you, if you don't know already, that at the lab, we have a program for high school research where high school students can join a lab and do cutting edge research. It's called Partners for the Future. If you're a high school junior, you can apply for this program in the spring. And then in your senior year, you can join a lab and do cutting edge research alongside one of our lab members. And this allows you to join any lab you like at the institute. It doesn't have to be plants. Also, we have programs, of course, in cancer biology and neurobiology. So please, if you're interested, please look out for that program, Partners for the Future. And with that, I'm done. I just want to really acknowledge the amazing people in my lab who've done this work. Actually, the work I talked about today was just most of the work of one postdoctoral researcher, Lei Liu, with a bit of help from my lab manager, Tara Skopelitis. And the others also have equally interesting projects that I'd love to tell you about some other time. And with that, I'd be very happy to take questions.
I muted. Thanks so much, Dave. That was really interesting.
Thanks.
And I'm starting to see some questions come through. So please, for people in the attendee section, if you would chat your questions to us so that we can answer them. The first one I'm seeing is thanking you for your presentation. I don't know if you're watching them as well, Dave. Can you sum up the research you've done so far in either a research question or a brief explanation? I wonder if it's saying, like, maybe in one sentence, how would you say what you do? Maybe that's what they're asking, but they could let us know if they want something more.
I mean, what drives me is figuring out how variation occurs in plants, why different plants look differently, and what are the genetic programs that control that, right? So so it involves finding the genes that control different aspects of development and then figuring out how they work. So I guess that wasn't one sentence, but so it's what's fun about it is that it involves a lot of different approaches. So there's genetics, but there's also biochemistry, there's computational biology, there's also microscopy, all sorts of different techniques, which makes it really exciting and collaborative. And it's really
wonderful. And again, we want to encourage the students to ask some questions, but I have another question, well, maybe one or two. One that I noticed, and again, knowing you for some time, but not knowing all of your background, is that there seem to be a lot of important women scientists who really helped you as a mentor. How would you say, the role, of making sure we have a diverse group of scientists coming up? Why is it so important? How did it impact you to have these wonderful mentors helping you in your career and getting you where you needed to go? Why is mentorship in that sense so important?
Yeah, thanks so much for bringing that up. It's also a really important issue and something that is very topical these days. That we need to talk about more, and I'm glad to talk about it. Yeah, I mean, I did have a lot of female mentors. I think it was probably just by chance. I mean, I didn't sort of seek out to have female mentors, but one thing that's really important is to have mentors who are really supportive of your research and very encouraging, and also who will sort of go to bat for you when you're applying for positions and things. So it's really important to have really good mentors. The general public sort of vision of a scientist is probably not a female. It's sort of an old white male like myself, but it's, we are trying very much to increase diversity. And we have programs at the lab to do that. And yeah, it's a very important area.
Second question I would ask, and again, invite people if they do have any others, because we're going to wrap up here in a moment and we'll be posting it. Well, there's a student question, so let me ask that one first. How did you find out about this particular area of research, and how did you figure out that you would really like to explain that? So I don't know if that might mean just meristems or corns, but how did you sort of get focused even on where you are today with that research?
Yeah, I think it's I like development because it's very visual, and I'm a visual person. I love art, and I love microscopes. So it's something that it's something that gives me a lot of chance to take beautiful pictures and to do that. But I think I got into that area really through my mentor, through my postdoc mentor, Sarah Haig, who and at that time this was an emerging area where not a lot of people were working. So I think if you're starting out in science, it's always good to look out for what you would call an emerging area, where some areas of science there may be already a lot of people working in them, and it may be harder to break into that field. But the area of development, using genetics to understand plant development, is something that kind of started around the time I was starting my career. So it seemed like a really good thing to go into.
And there's another question here, although it's from one of our co-panelists who's behind the curtain, so to speak, but it's a great question, which is, you mentioned the Partners for the Future, which again, we'll maybe post, when you put this up on YouTube, post a link to that so people can find it. But in general, if somebody is interested and maybe they're not close, because people may be watching this from a distance, maybe not close to Cold Spring Harbor, what's the best way to approach a scientist that they might be interested in as a high school student, of finding out if there's a position in the lab or some way to learn more about their research, sending an email? What do you think is good for you? Because I'm sure you get lots of requests.
Yeah, I think it's difficult because I people do get a lot of requests. I think you have to be persistent and patient, and try to do a little bit of research on the person's on their research before you write to them, so that you can say why you're interested in their research, and give some background into what you want to do. Obviously right now, because of COVID, none of us are really taking extra students in our labs, but generally I take a lot of summer students. I take students during the year. So it is definitely possible to get a position in a lab, and there are lots of opportunities in the New York area, not only Cold Spring Harbor, but there's a lot of great research universities around. So yeah, just be persistent and expect not to get a response if you write to somebody, because everybody gets way too many emails. But usually what I do with emails is I recommend you write an email, and if you don't get a response, just send a reminder a few days later, and that usually helps. Sort of for me at least that helps. If I forget, I usually respond the second time.
All right, we've got one more question, and then we'll just call it a wrap. It's an interesting one. So how do you reflect on failure? For example, if you didn't get the results you hypothesized in your research, how do you deal with that?
Yeah, it's a really good one. I mean, basically, we often don't get the expected results. So one thing is you can try and turn a negative result into a positive. So if you don't get the expected result, try to figure out why. And it may be telling you something interesting, if the system isn't working in the way you expected. Sometimes we have ideas about how things work, and there may even be things published about how things work, and they could be wrong. So, sometimes a negative result can turn into a new area of research. I mean, there have been many examples of that. So, but I think the key is to not get too distracted by that. Sort of try to balance it so that you keep and usually most people are doing several sort of different lines of research in parallel. So if one comes to an end, or you can't figure it out, then usually another one will give you something to work on. So yeah. So
yeah, if you really believe in a hypothesis and it's not working, that means there's
Mhm.
more to understand, and you're going to have to
Exactly.
keep
Yeah. We're never finished. And I mean, the other great thing is that, there's so much new technology coming along all the time that even now, questions we can answer now, I wouldn't have dreamed of attacking 5 years ago, like CRISPR and next generation
Exactly.
sequencing. It's just, it's amazing that the resources, the opportunities it gives you to answer different questions.
Well, wonderful. Well, thank you for your time, and also thank you to all the attendees. And we hope that you'll be back with us for the next speaker is going to be on December 10th with Dr. Dos Santos. So spread the word, invite people, and thank you so much for your questions and for your time, Dave. It was a great presentation.
Okay, thanks so much. Have a great
Thanks,
evening.
everyone. Bye.